Hello friends, today in this video we are going to discuss about the MCQs on immunology with detailed explanation, and this is the 15th part of the series of more than 1800 MCQs on immunology, and this will be beneficial for many life science exams like GATE or NET, CSIR NET, or any kind of nurse exam or hospitality exams, or also it can be beneficial for NEET exams. Okay, so move on to the first question answer session of immunology. So the following is an isoantigen. So HLA, the options are HLA, pollen grains, sheep and ox red blood cells, or thyroglobulin, and the right answer about the isoantigen is option A. So HLA is the isoantigen. So isoantigen is an antigen existing in alternative or allelic forms in a species, thus inducing an immune response when one form is transferred to members of the species who lack it. So typically isoantigens are the blood group antigens. Okay, so move on to the next question, that is counter immunoelectrophoresis is electrophoretic separation of antigen or simultaneous electrophoresis of antigen and antibody, or single diffusion in two dimensions or double diffusion in two dimensions. So the right answer about the counter immunoelectrophoresis is option B. So it is simultaneous electrophoresis of antigen and antibody. So counter immunoelectrophoresis is a laboratory technique used to evaluate the binding of an antibody to its antigen, and it is similar to immunodiffusion but with the addition of an applied electrical field across the diffusion medium, as usually an agar or polyacrylamide gel. The effect is rapid migration of the antibody and antigen out of their respective wells towards one another to form a line of precipitation or a precipitin line indicating binding. Okay, so here the antigen is present, here the antibody is present. So when we start the electrophoretic current in this way, the antigen will go this side and antibody will go this side, and here in the middle they produce a precipitin line. So this is the process of counter immunoelectrophoresis. Okay, that is a antigen antibody complex test. Okay, so move on to the next question, that is Ascoli's thermoreceptin test is slide flocculation test or tube flocculation test or ring precipitation test or none of the above, and the right one is option C, that is ring precipitation test. So Accol's thermoprecipitin test used to identify anthrax bacilli in animal hides and meats, and this test was designed to detect Bacillus anthracis antigens in the tissue of animals. So the reaction is demonstrated by layering antigen solution over the column of antibody in a narrow tube. The precipitate forms at the junction of the two liquids like this. So here antigens are present, here first antibodies are added. So here in a zone of equivalence visible precipitation is done. Okay, so this is also a antigen antibody complex test. So move on to the next one, that is coagglutination test detects negatively charged antigens from from body fluids or depends on presence of protein A of Staphylococcus aureus or detects low molecular weight substances or is a immunoassay depending on enzymatic activity. The right answer is option B, that is co coagglutination test depends on presence of protein A of Staphylococcus aureus. So coagglutination test, basically it is also agglutination test in which inert particles like latex beads or heat killed Staphylococcus aureus Co11 strain with protein A are coated with antibody to any of a variety of antigen and then used to detect the antigen in specimen or in isolated bacteria. So here the antibody attached with the latex bead or S. aureus Co1 stain with protein A, and then we add a specific antigen, they bind to it. That is known as a coagglutination. Okay. So move on to the next question, that is B lymphocyte has surface immunoglobulin or has receptor for FAB portion of IgG or is involved in phagocytosis or forms SRBC rosette, and the right answer is option A, that is B lymphocyte has surface immunoglobulin. So B cell, also known as B lymphocyte, are a type of white blood cell of the lymphocyte subtype. So they function in the humoral immunity component of the adaptive immune system, and these B cells produce antibody molecules which may be either secreted or inserted into the plasma membrane, where they serve as a part of the B cell receptor. Okay. So move on to the next, that is macrophages has surface immunoglobulin or has receptor for FAB portion of IgG or is involved in phagocytosis or forms SRBC rosette, and the right answer is option C, that is macrophages is involved in phagocytosis. So macrophages are a type of white blood cell of the innate immune system that engulfs and digests pathogens such as cancer cells, microbes, cellular debris, and foreign substances. Okay. So next question is highly sensitive test gives false positive, false negative, or high positive predictive value, that is PPV, or low negative predictive value, that is NPV. And the right answer about the highly sensitive test gives option A, that is false positive. So in general, high sensitivity tests have low specificity. So in other words, they are good for catching actual cases of the disease, but they are also come with a fairly high rate of false positive. Okay. So highly sensitive tests give false positive test mostly. So next one is highly specific test gives false positive, false negative, low PPV or high NPV, and the right one is option B. So highly specific test gives false negatives. So the test specificity is its ability to correctly indicate when an individual is not infected or doesn't have antibodies. A test that is very sensitive is less likely to give false negative result, and a test that is highly specific is less likely to give false positive. So false negative, the patient have the disease but the test is negative. So the sensitivity of a clinical test refers to the ability of the test to correctly identify those patients with the disease. A test with 100% sensitivity correctly identifies all patients with the disease. Okay. So the next question is gram-negative organisms produce endotoxin or septicemia leads to disseminated intravascular coagulation or both of the above or none of the above, and the right answer is option C. So both of the above. So gram-negative are both of the above, that is organisms produce endotoxin and septicemia. Okay. So septicemia is a serious bacterial infection that affects the blood. So disseminated intravascular coagulation, DIC, or disseminated intravascular coagulation or DIC is a complicated condition that can occur when someone has severe sepsis or septic shock. Both blood clotting and difficulty with clotting may occur, causing a vicious cycle. Okay. So the next one is gram-positive organism produce exotoxin or septicemia lead to disseminated intravascular coagulation or cocky are always non-motile or all of the above. The right one is option A. So gram-positive organisms produce exotoxin. Okay. So exotoxin means they are produced inside the cell but they act on outside of the cell. That's why they are called exotoxin. So tuberculin allergy can be passively transferred by serum, lymphoid cells, both of the above, none of the above. The right one is option B, that is lymphoid cells. So tuberculin allergy can be passively transferred by lymphoid cells. Okay. So tuberculin type is when small dose of tuberculin is injected intradermally in an individual sensitized to tuberculoprotein by prior infection or immunization, and indurated inflammatory reaction develop at site within 48 to 72 hours, and in unsensitized individuals no response. So useful indication of the state of DH to the bacillus. So here this is the addition of the tuberculin to our body, and after reaction it gives, if it is positive then it gives rise like this. Okay. So, and what is lymphoid cells? Lymphoid cells means move around the body in the lymph node and act more slowly to kill more specifically. So lymphoid cells are T cells, B cells, or natural killer cells. Okay. So the next one is immune complex disease is type 2 hypersensitivity or cause massive complement activation, or both of the above, none of the above. And the right one is option B. So immune complex disease causes massive complement activation. Monoclonal antibodies are produced by hybridoma technique or produce antibodies against a single antigenic determinant, or are very useful tool for diagnostic and research purposes, all of the above. And the right one is option D, that is all of the above. So monoclonal antibodies are all of the above. Okay, so move on to the next, that is interleukin 2 is a powerful modulator of immune response or activates B cells, or both of the above, none of the above. So interleukin 2 is a powerful modulator of immune response, that is option A. So interleukin 2 is a interleukin, a type of cytokine signaling molecule in the immune system, and interleukin 2 is part of the body's natural response to microbial infection and is discriminating between foreign, that means non-self, and self, and the major source of interleukin 2 are activated CD4 plus T cells and activated CD8 plus T cells. Okay, and interleukin 2 has essential role in key functions of the immune system, tolerance and immunity, primarily via its direct effect on T cells. In the thymus, where T cells mature, it prevents autoimmune disease by promoting the differentiation of certain immature T cells into regulatory T cells, which suppress other T cells that are otherwise primed to attack normal healthy cells in the body. So this is the role of interleukin 2, to regulate the T cells to cause the autoimmune disease. So move on to the next question, that is atopy is mediated by IgE, caused by inhalants, or caused by ingestions, or all of the above. And the right one is option D, all of the above. So atopy is a specific category of allergy. It isn't a single disorder but rather a way to describe a group of disorders. So atopy conditions are often the result of your immune system overreacting to some triggers. So they are part of a group of conditions known as type 1 hypersensitivity disorder. Atopic conditions are not uncommon. Some of the most frequently diagnosed atopic conditions include allergic asthma, allergic rhinitis, atopic dermatitis, or allergic conjunctivitis or food allergies. So during atopy, your body responds to an otherwise harmless or mild antigen by making too much of a specific antibody called immunoglobulin E. So this leads to the release of histamine and other chemicals that cause inflammation. So next one is precipitation reaction can be converted to agglutination reaction by carrier particles like latex particles or bentonite or red blood cell, or all of the above. And the right one is option D, all of the above. So precipitation reaction can be converted to agglutination reaction by carrier particles, because the main difference between precipitation and agglutination is their nature of antigen. So in precipitation, the antigen is soluble antigen. In agglutination, the antigen is insoluble antigen. Okay, so move on to the next, that is B lymphocyte predominate in gut associated lymphoid tissue or originate from bone marrow or is equivalent to bursa fabricius in birds or all of the above, and the right answer is option D, all of the above. So B lymphocyte predominate in gut associated lymphoid tissue, originate from bone marrow, and equivalent to bursa fabricius in birds. So move on to the next, that is collection of convalescent phase serum sample after acute phase serum sample is done after 4 to 5 days, 7 to 10 days, 2 to 3 weeks, 1 month. So answer is option B, that is 7 to 10 days. So basically what is convalescent serum? So serum obtained from one who has recovered from an infectious disease and considered to be especially rich in antibodies against the infection agent of the disease. So convalescent phase serum sample was defined as a serum taken 10 days or later after the onset of rash. So collection of convalescent phase serum sample after acute phase of serum sample is done after 7 to 10 days. So next one is advantage of PCR is or are, that is detects organisms which are slow growing, non-viable, non-cultivable or all of the above, and the right answer is option D, all of the above. So PCR means polymerase chain reaction is a technique to make many copies of a specific DNA region in vitro in a test tube rather than an organism. Okay, so next one is disadvantages of PCR is or are interference by certain inhibitory substances or selection of organisms towards which primer are targeted or non-availability of culture subsequently for characterization or all of the above, and the right answer about the disadvantages of PCR is option D, that is all of the above. So next question is PCR is a highly sensitive test or avoid delays associated with culture or is superior to hybridization in sensitivity or is all of the above, and the right answer is option D, is all of the above. So next one is molecular methods are highly specific, not ideal to detect mixed infection or highly sensitive or have interfering substances in the specimen, and the right one is option A, that is are highly specific. So move on to the next, that is food allergy is associated with all the following foods except sea fish, prawn, milk or meat, and the right one is option D, that is meat. And the next one is food allergy is associated with nuts, eggs, mushroom, all of the above, and the next one is answer is option D, all of the above. So food allergy is associated with nuts, eggs, mushrooms. Okay, and the last question of this video is dust allergy is associated with pollen grains, animal danders or house dust of ragweeds or all of the above, and the right answer is option D, that is all of the above are dust allergy. Okay, so dander is material shed from the body of humans and other animals that have fur, hair or feathers. Okay, so these are all about this video about the immunological MCQs. So thank you for watching this video and we will see you on our next video, that is the part 16 of immunological MCQs. Okay, so thank you for watching this video.